Hey everybody, this is going to be the review for Learning Target 7B, which says I can use and analyze Punnett squares to calculate the possible phenotypic and genotypic ratios of offspring in a dihybrid genetic cross. And how you need to be able to do that is I can explain how genotypes or alleles are expressed as phenotypes in living things using a dihybrid Punnett square. I'm going to start right now by saying if you don't understand any of summative 7A learning target, so if you don't understand 7.2, 3, or 4, then you need to go back and you need to watch the Learning Target 7A review video. Because if you don't understand this, then none of what we're going to talk about in this video is going to make any sense. So again, if you don't know any of these, go back and watch the first video and then come back and watch this one. All right. So this learning target basically just wants us to be able to do a dihybrid cross. Now, I want to remind you that dihybrid crosses always have 16 boxes. So a monohybrid cross has 4 boxes and a dihybrid cross has 16 boxes. When we look at dihybrid crosses, we're looking at 2 genes together. So in this example, we're looking at a genotype for 2 parents, and the genotype is the same. We are looking at whether the plant is tall or short, and whether the plant is purple or white in the same instance. So these 2 genotypes are heterozygous for both traits. The first step into solving any of these problems is to FOIL your genotypes. You have to get the gametes on the top and the bottom. The gametes are going to be 2 letters. They're going to be one of the first gene and one from the second gene. So this can't be T, big T, big P, little p. That's incorrect. I have to have one of each. I have to have a T and a P in each one of these boxes. But the question is, which T and which P? Because there's 2 different alleles. There's 2 different T's and there's 2 different P's. So the first way you can do this is you can use this FOIL method, and that stands for first letters, outside letters, inside letters, and last letters. So if I was to do that, I would take the first letters of the genotype, so big T, big P, and I would put them right here. Outside letters is big T, little p, and I would put those right here. Inside letters, little t, big P, in the third box, or last letters, little t, little p. That's the FOIL method. Another way to do this is to distribute. This is how I said that I do it, and this is how I've been doing all the examples with you. I take the first T and I give it to the first P, and I would put it right here. Then I take the first T and I give it to the second P. That would be my next one. Then I'm going to take the second T and give it to the first P for the third box. And for my last box, I'm going to take the second T and give it to the second P. If you're somebody who remembers numbers, the way to remember this is 1 to 3, 1 to 4, 2 to 3, 2 to 4. If you're not somebody that remembers numbers, don't even worry about it. That's just another way to help you remember. The last way to do this is to take these 2 genes and put them in a monohybrid cross. So I would make a box and I would put big T, little t on top and big P, little p on the side. And then I would cross them like a normal monohybrid cross, and that'll give me all of the alleles to put in these boxes. So down here you have the FOIL method. It's showing you which letters would be in each genotype. Once I figure them out, I'm going to put them in the boxes on top, and then I'm also going to put them on the boxes on the left because these 2 parents have the exact same genotypes. Now what I'm going to do is I'm going to take the letters on top and bring them down, and I'm going to take the letters or the alleles on the left and bring them over, just like in a monohybrid cross. Important to remember that T's are going to go with other T's and P's are going to go with other P's. So when I bring these two together, it's going to be T, T, P, P, keeping those similar alleles together. And these are all going to be capital because both of these two alleles are capital on this side. I'm going to follow that same pattern. Bring what's up top down and what's to the left outside to the right and slide it over until I have every single box filled out. I would always stop at this point and double check your work. Make sure that you didn't accidentally put a capital P where there's supposed to be a lowercase p. You know, make sure that you're very thorough with this. These can get a little bit confusing. The next step, once I've had these all filled in, is to interpret this. So what does this all mean? Not only am I looking at whether you're tall or short, I'm looking at whether the flowers are purple or white, but I'm looking at them at the exact same time. So they're never going to ask you just how many of them are tall, which would be just this, or how many of them are short, which are these ones over here. They're never going to ask you which ones are just purple and which ones are just white. These would be the purple ones and these would be the white ones. They're always going to ask you what percentage of the next generation are tall and purple, or tall and white, or short and purple and short and white. That's the whole point of a dihybrid cross. You're looking at two things at once. So this question says, how many boxes show a genotype that will express a tall plant that also has purple flowers? Well, I know tall is dominant, so anything with a capital T, and purple flowers. Purple is also dominant, so anything with a capital P is going to be what I'm looking at. So when I get this question, how many of the boxes or how many genotypes show a purple plant that's tall, I'm going to look for big T's and big P's, and I'm going to count every box that has a capital T and a capital P. So once I do that, I realize that 9 out of the 16 will be tall and purple. The next question says how many boxes show a genotype will express tall and white flowers. So now I'm looking for capital T's and 2 lowercase p's because in order to have a white flower, it has to have the recessive genotype. So only 3 out of the 16 are going to have that specific genotype.